All right, hello everyone. Welcome to the NIH Complementary Nouns Reduction to Practice Challenge webinar. Thanks for taking the time to join us today. Please feel free to say hello uh, in the chat. Let us know where you're joining from. Our agenda for today. All right, we're going to be providing an introduction of our panelists, and then we're going to have an overview of the challenge along with discussing how to enter and submit your solution, and then we're going to move into the Q&A for today. Just a reminder, please do submit your questions in the Q&A box you see on your screen there. I may not get to all of your questions today on the webinar. For those that we don't get to, we'll look to respond to via the challenge page. We're also going to be uploading a recording of today's webinar to the challenge page. So I could start off with introductions. I'm Andrew Osansky. I'm a project manager here at HeroX, and we're partnered with the NIH to administer this challenge. I'll now pass it over to Margaret to begin introductions on the NIH side.
Thank you very much. So I am Margaret Ochochinska, and I'm a program leader in the Common Fund in the Office of Strategic Coordination within NIH, and I'm helping to coordinate efforts in the Complement Animal Research and Experimentation Complementary Program. Joining me today is also Christine. Hand it over to her for introduction.
Hello, my name is Christine Happel. I'm a program officer in the Office of Special Initiatives at the National Center for Advancing Translational Sciences, or NCATS. And with that, I will hand it over to my colleague, Dr. Thad Shug.
Hi, everyone. I'm Thad Shug. I'm a program director as well. I work at the Population Health Branch at the National Institute of Environmental Health Sciences. Margaret?
Thank you so much, Christine and Thad. So with that, I wanted to also welcome everyone to the webinar, and I'm happy, and my colleagues and I at NIH are happy to welcome you to the Reduction to Practice NAMS webinar, and we're excited to invite you to build the future of human-based biomedical research with new approach methodologies. And so we will briefly go over some background next. Next slide. So just providing a bit of background on the Common Fund, which is where this program was spearheaded in coordination with the NCATS and NIEHS. And so essentially the Common Fund was built out of the NIH roadmap back in the early 2000s, and it was launched. And in 2006, Congress unanimously reauthorized the NIH and established a Division of Program Coordination, Planning, and Strategic Initiatives, or DPCIPSI, within the Office of the Director. And the NIH Common Fund was created to provide a dedicated source of funding to enable cross-NIH research. Next slide. And so the purpose of the Common Fund is to move the NIH mission forward faster to support bold scientific programs that catalyze discovery across all biomedical and behavioral research, to advance areas of biomedical and behavioral research important to the missions of multiple NIH institutes and centers, and to spur subsequent biomedical advances that otherwise would not be possible without an initial strategic investment. Next slide. And so some of the features of these programs and how they can catalyze biomedical discovery are they can accelerate emerging science, remove research roadblocks, enhance the research workforce, and support high-risk, high-reward science. And this challenge prize is one of those mechanisms that we can utilize to enable this catalytic activity. Next slide. And so just in summary, the Common Fund science and management are collaborative. So scientists from various disciplines provide input as we plan new programs like this complementary program that the challenge is a part of. We also enable researchers to participate in interdisciplinary consortia to tackle shared goals. And leadership and staff across the institutes and centers at NIH and across the federal family work together to design funding opportunities and oversee the projects, such as this challenge that we are discussing. So with that, I would like to turn it over to Christine to tell you more about the complementary program. Thank you.
Thank you, Margaret. So Complement Animal Research and Experimentation, or complementary, is a new Common Fund program. And the purpose of this program is really to catalyze the development, standardization, validation, and use of human-based new approach methodologies, or NAMs, that will transform the way we do basic translational and clinical science. So the overarching goals of the complementary program are to better model and understand human health and disease outcomes across various populations, to develop NAMs that provide insight into specific biological processes or disease states, to validate mature NAMs to support regulatory use and standardization, and to complement traditional animal models and make biomedical research more efficient and effective. Next slide. So NAMs can generally be broken down into three main categories. We have our complex in vitro models. These can be used for things like emulating population diversity. We have our in silico models that can simulate things like healthy and diseased individuals, and our in chemical models that can capture dynamic changes such as epigenetic changes, changes in biochemical pathways, or chemical genetic changes. And then we have our last phase is our combinatorial NAMs. Combinatorial NAMs generally incorporate two or more of the above NAMs categories. And really the goal with these combinatorial NAMs is to integrate these different NAMs elements into a synergistic approach that augments the gaps or deficiencies in the individual NAMs approaches. So while NIH has supported a number of activities in the NAM space, these have been in the development of in vitro systems such as microphysiological systems, 3D organoids. NIH has supported activities such as the development of digital twin models and even in chemical screenings with the Tox-21 program. However, the goal of complementary is really to develop and integrate novel, innovative NAMs with sufficient biological complexity to answer key questions in human biomedical research. Next slide. So what we're focusing on today in today's webinar is the Complementary NAMs Reduction to Practice Prize Challenge Competition. So this challenge aims to bring forward early stage combinatorial NAMs to a practical and usable form within a 3-year time period. This challenge will take place across 3 phases. It'll be 3 years in length, and it has a $7 million in total prize money. The phase 1 submission period has already opened on September 30th of this year and will remain open until March 1st of 2026. Winners are anticipated to be announced in July of 2026. Next slide. So with this slide, you'll see the structure of our NAMs Reduction to Practice Challenge. It has been structured as a funnel. So right now we are in the first phase, and we have put out an open call for solutions for the development of these combinatorial NAMs that can address deficiencies in human health and modeling. Phase 1 applications are due, as I said, by March 1st, 2026. And after that, there'll be a selection process where we anticipate up to 20 winners being awarded $80,000 each. Following phase 1, we will then move into phase 2. There'll be 2 different deliverables within the phase 2 portion of the challenge, each with distinct winners. With milestone 1, we anticipate 10 winners. In milestone 2 for phase 2, we anticipate 7 winners. Winners within phase 2 will then be able to move into phase 3 of the challenge and collaborate with the validation and qualification network that we have established within the complementary program for independent testing and validation. More information about the structure and the different phases of this program can be found on the HeroX website. So we're not going to go through that in depth today, but this is the overarching structure of the challenge competition. Next slide. Also within the challenge, we do have a number of priority areas that include chronicity, neurobiological models, personalized health. Next slide. As well as cross-disease pathogenesis. So this includes platforms that can look at multiple diseases at a time, or platforms that may be broadly generalizable across a number of different diseases. Priority areas also include toxicology and safety, as well as human health and protection. Next slide. 2 of the common questions that we get for This program is what is a combinatorial NAM solution? So combinatorial NAMs are ones that integrate NAMs technologies across modalities, so either in vitro, in chemico, or in silico, or it can also include distinct NAMs technologies within a modality. The overarching idea is that we're trying to create a cohesive solution that leverages multiple methodologies for a comprehensive insight into human biology. The second question we often get is, how mature must a NAM be to be responsive to this challenge? We think NAM submissions should be of a sufficient technical readiness level to be ready for validation and qualification within a 3-year time frame. Is our general advice. And I think with that, I will ask for next slide, and I will pass it off to my colleague, Dr. Thad Shug.
So here's the timeline of the complementary RTP challenge. We're at the 12/10 informational webinar stage. We released the challenge. It was launched at 9:20. The submission deadline is March 1st. And again, the awards will be announced at the end of July 2026. Next slide, please. So to participate in this competition, you can come in either as a team or an entity. So if you come in as a team, you choose a captain who must be a U.S. citizen or a permanent resident. If you come in as an entity, you'll identify a point of contact, and the entity must be incorporated and located within the United States to be eligible. Next slide, please. So note about using federal funds to compete. So innovators cannot use federal funds for the development or support of their proposal unless use of the federal funds is consistent with the terms and conditions of the contract, grant, or cooperative agreement federal fund award. If that is the case, then federal funds will be used as program income, and that'll be that is stipulated in the terms of the award. So next slide, please. So the proposals will be evaluated in 5 different evaluation criteria. First, the approach. Is your NAM proposal, does it clearly clearly identify your plan? Is it will it lead to validation? Does it incorporate best practices, account for potential roadblocks, and what is the likelihood of success? For the team evaluation, it's an interdisciplinary evaluation of the scientists and participants from your team and their experience. Feasibility of approach, is evidence provided to demonstrate that the context and use of your NAM and the quality and quantity of your preliminary data support your NAM development. Innovation, is your tool novel and technically innovative? And then finally, potential impact. Will your NAM, what's the value of your NAM to support the 3 R's: reducing, refining, and replacing animal use? And is your NAM going to up for validation and use in the marketplace? So those are the evaluation criteria. Next slide, please. So here's the slide that explains the submission process. So on the HeroX website, you'll find all this information. Where you click on that link, you can come in again as a team or an entity, where you would identify yourself and then provide the information on the submission requirement pages. The submission length of these pages and the criteria are well spelled out on the submission website. So that'll walk you right through the submission process. And again, in order to submit, you just click this blue button here on the left side of the screen that says, can barely make it out, but It is basically you click that button to submit your application. Next slide, please. So that's it for the slides, and we're on to questions.
Great. Thank you all for providing that overview. And I'll just add the challenge page has all the information that you'll need, and you can go ahead and register and solve the challenge to make your submission through there. There's also the forum section. If you do have any questions that come up following this webinar, please feel free to post them there. You'll also see on your screen there's an email as well where you can submit questions also. All right, so we are now going to move into our Q&A portion. Just a reminder, there is a Q&A box on your screen. Please go ahead and submit your questions there, and then we will ask them to our panelists here. All right, so getting us started, first question, could you define too broad and too narrow with regard to scope?
All right, I
Go
can
ahead, Christine.
try to attempt that one. So that's going to be a tough one because there are no parameters set for what we're looking for in terms of scope. I think that obviously there we are requiring a combinatorial NAM. So that is going to be one of the requirements. I think the best place to look will be the judging criteria, which are outlined on the HeroX webpage, and you'll notice that the criteria are not all weighted equally. There are different weights for some of the criteria as listed on the website, and that's likely going to be the best place to look, but we do not have any specifications in terms of scope that we are looking for. I don't know if others have anything to add.
Yeah, I agree with Christine on her answer. And your project must be feasible within the time window of the three phases, which is a 3-year window. And the evaluation criteria give you a good idea of how ambitious your project can be, whether it meets those standards that you're being evaluated on or not.
Excellent. Thank you. Next question. Can you discuss the kind of support that will be provided for companies who work in NAMs throughout the challenge?
Yes, I can comment briefly on that as well as the HeroX team. So there will be additional support for the second phase. So after the initial phase in preparation for phase 2, there will be extensive support provided in terms of potential next steps for delivery, for implementation, for adoption, and for criteria for entry into the validation qualification network, so that there will be additional support provided. But I'll also let the HeroX team chime in. Others, thank you.
Yep, I think
Thank
you
you.
covered it all there. Yeah,
Yep.
and I think just that we can add is that there will be check-ins with the team as well. So you will be supported throughout follow-on phases. All right, next question. Can researchers working in nonprofits apply for this challenge?
Yes.
All right, that's an easy one there. Can you describe how submissions of proposals differ from traditional NIH mechanisms?
Yes, I can take that on. The difference is that while traditional mechanisms are what the investigators intend to develop, while challenges are to award prizes for a solution that has already been completed. So essentially, you're being evaluated on what you have developed at that stage and if it is what we're looking for. So that's the primary difference in compared to other mechanisms.
I agree with Margaret. I'll just add that the submission is different because the submissions for this challenge will be through HeroX website, as indicated in the webinar, and then they'll be evaluated differently, not within the peer review system that traditional NIH grants and cooperative agreements go through, but in a objective review process that utilizes federal experts in the NAMS space.
Excellent. Thank you. And just a quick reminder to everyone on the webinar, if you do have some questions, please put them into the Q&A box. We'll be happy to ask them to the group. All right, moving along. Next question, would technology that aims to reduce the requirement for endpoint histology animal models be in the spirit of NAMS?
Yes, it would be in the spirit of the 3 Rs. So, yes.
Excellent. All right, next question. Do you need NAMs that can eliminate animal testing or just reduce animal testing?
I'll say neither is a requirement. However, we are one of the goals of the complementary program overall is to reduce animal testing. So that is something that would certainly be advantageous to any proposals.
Excellent, thank you. Is there an advantage to meeting multiple priority areas, or are we expected to propose into one priority area?
I can take that
So
as,
there are, go ahead, Margaret.
no, go ahead, Thad.
I was just going to say there's priority areas that are listed on the application page on the HeroX website. When you click to submit your proposal or to fill out your information, there's priority areas that are clearly identified that NIH is looking for.
Excellent. All right. Do you have any specific requirements or standardized experiments to be validated across all the proposed technologies?
We have not outlined specific requirements at this time. Right now, during phase one, we have an open call as groups, winners are announced from phase one and move into phase two. We will announce more specific requirements for phase two and then down the line for phase three as well. However, it will generally encompass things like intralab, reproducibility testing. So there'll be broad categories, but they will not be specific experimental requirements.
Great. All right. Next question. Validation is a key component for NAMS development. This may require the use of animals due to ethical or technical constraints in performing validation on purely human platforms. Is animal use allowable in the development phase?
No, animal use is not allowable for this challenge.
All right. Next question is, are there any requirements for specific disease models? How do you value the weights of disease types, importance versus the robustness of the NAMs?
There are not specific disease models that are included or excluded, but there are priority areas, again, which are identified on the HeroX website where the application form is located.
Great. All right. Given the VQN is still in the early stage of development, do you think it will be ready for support for this challenge, especially in phase two?
So We envisioned that so the VQN is being developed, as was mentioned by the question. What we are saying is that the HeroX will support in collaboration with additional membership for advising on the projects that are being developed through this challenge. The end goal is for the VQN network to consider some of these applications and solutions for entry into the VQN in the third year. So the support that we are talking about is the development of those proposals, the feasible proposals and solutions within the 3-year time frame, but the entry will be towards year 2 and 3 to the VQN. So at that stage, we envision having additional support through that network and the subject matter experts there.
Excellent. All right. Question, can you define validation?
Sure. This is definitely one of those terms that can be interpreted in multiple different ways. Currently, we are using the FDA definition of validation, which means that validation is a process of establishing documentary evidence and demonstrating that the procedure, process, or activity carried out maintains the desired level of compliance at all stages, and it will consistently produce the expected results. So it's a very broad definition of validation.
Excellent. And just another reminder to everyone, please do submit your questions to the Q&A box and we'll be ready to ask them to the group. All right, next question. Can an entity be involved in multiple applications?
Yes, if the applications are unique, so the applications themselves, the solutions don't overlap and don't cover the same area being proposed.
Great. Next question, can you provide any insights into who the end user would be for a NAM?
Can take that and then others feel free to chime in, but essentially the end users here is essentially the stakeholders that are part of the validation qualification network and future networks. So it would be a range of private sector entities and public sector entities. So it's really depending on the context of use. It could be on the regulatory end, it could be on the biomedical research side. So it's a pretty broad area as well here.
Excellent. Next, would the program require prefer project team to meet all three in vitro, in silico, and in chemical platform, or one or two of these areas is fine?
Groups are not required to meet all three different categories of NAMs. We would prefer to see at least two. And the reason for that is because we're really trying to augment deficiencies in each of the individual NAMs types.
All right. Next question. What level of details required for the project? Do you require the design of the proposed method?
I will take that question and let others chime in as well. But essentially, so there's certain character limits for the submissions, but within the context of just key descriptors, as much information as you can include, so that we can assess so that the reviewers can assess the feasibility and the impact of the proposal and the likelihood of it being completed within the 3-year time period.
Great. All right. Next, can you further define two distinct NAM technologies within a modality?
So this could be a combination of different like in silico methods, resulting in something like a digital twin modality. So combining artificial intelligence, machine learning with another other in silico methods. So that would be one way to combine different um, NAMs within a single modality.
Excellent. All right, next question. Are invertebrate small model organisms considered NAMs under this program?
They're not considered NAMs under this program. We do allow for legacy data to be used for some of the proposals, though. So just a previous question about animal models in general, that is allowable. But for the actual projects, we do not consider those NAMs within the purview of the complementary program.
Great. All right. Are ex vivo models allowed, for example, lung slices?
Yes, they are allowed.
All right. Next question. Is it required that we have our own preliminary data?
It is not required. Preliminary data is not required, but groups will be a better position to show that they have some preliminary data and are capable of putting together the prototype NAM as it progresses within the challenge. So generally, we're going to be looking for NAMs that are likely, already established and probably around the technology readiness level of about 4. So that way they'll have sufficient preliminary data and but entering into the challenge and then continue towards validation efforts as we move through the 3-year timeline of the challenge itself.
So the feasibility and approach criteria would be used to judge whether your NAM is likely to be proficient or not, and a sufficient quantity and quality of preliminary data would support that much stronger.
Great. Next question. Can the application be limited to one context of use or should it propose multiple?
And focus on one or additional ones. There's not a preference, just again for feasibility and impact of the proposed solution.
All right. Next question. Is NIH exploring any other approaches beyond crowdsourcing to solve this problem?
Yes, we are. In fact, the entire complementary program has 3 major pillars. So we're utilizing cooperative agreement mechanisms for technology development centers. We're also standing up a NAMS data hub and coordinating center. We are also establishing the validation qualification network. So this challenge is one of many types of approaches that we're using for answering or developing new alternative methodologies. NIH as a whole also has a focus on human-based methodologies, and a roadmap per the FDA roadmap is being co-developed in this space. So there's multiple avenues to answer this question, but we hope that the challenge mechanism is one nimble approach to answer it as well.
And I'll also add that NIH has a robust small business innovation program that most institutes support that also have a number of NAM type projects that are supported.
Excellent. Thank you. All right, next question. Do you prefer a tightly defined context of use, or are you open to broader platform contexts of use that span multiple indications as long as the milestones and validation plan are clearly defined?
I can take that. So while there is no requirements in terms of the context of use, we generally find that it is advantageous to have a single, more well-defined specific context of use as these NAMs move further along into the validation efforts.
Excellent. All right. Next question. What approaches are within scope for this challenge and what approaches are not of interest to NIH?
I think we answered a similar question, so I would direct you to the priority areas. So in terms of the focus areas, but the range of approaches, I'll leave it up to the solvers, but just to ensure that you have highlight that your proposals touch on some of these priority areas.
I'll just add to that that these are all should be human-based NAMs. So animal-based NAMs would be out of scope for this competition.
All right. Next question. Could you please elaborate more on the required in silico component? As an example, how complex, simple should it be?
I'm sorry, could you read that one more time?
Yeah, certainly. Could you please elaborate more on the required in silico component? And as an example, it has how complex, simple should it be? Complex slash simple. So,
So there is no requirement for an in silico model. It is up to the solvers to define the type of combinatorial NAM that they would like to propose. So we really kept the NAM types very broad and up to the technology developers in order to really get to the NAMs that not only are technically more advanced and ready for validation efforts, but that are also going to be most impactful in terms of the human biology in which they're trying to model. I don't know if others have anything to add to that.
I think that covers it. Thank you, Christine.
excellent. Thank you. And just another reminder to please put your questions into the Q&A box. We just have a few more here, so please do add them and we'll ask them to the panel. All right, next question. Can you provide more detail about your vision for phases 2 and 3 of this challenge?
Sure. So I can start off and others can chime in as needed. So following the announcement of winners from phase 1 of the program, those groups will then move into phase 2, which is broken down into 2 milestones. So for milestone 1 of phase 2, we'll be looking for progress towards performance and testing requirements, showing that the DAMS platform can be used for a specific context of use. We'll also be looking for construction and scaling up of the NAMS platform to get to a prototype that will be fit for purpose to move into the later stages. So that will be for milestone 1 of phase 2. Milestone 2 of phase 2 will then be looking for deliverables such as progress towards internal validation of the NAMS platform using things like reference standards, compounds, or agents, whatever's needed to demonstrate within laboratory reproducibility. It'll be looking for documentation of the not only the platform itself, but of the data that is being generated. And then it will also look for collaborative efforts between the solver and the validation qualification network, as well as the data hub and coordinating center. After milestone 2, in phase 2, there will be, we anticipate, I think, 7 awards being made at that time. And then those solvers will then move into phase 3, which is really prototype delivery and validation qualification by an independent body, which will be the Validation and Qualification Network that is being stood up as part of the complementary program. We anticipate that this is, that portion of the program, phases 2 and 3, will be about another 2 years after the program. I think the most important thing is that all solvers, though, must enter during this phase 1 to be able to progress to the later phases. So there will, after this phase 1 portion, there will not be additional entry points into the challenge funnel structure.
Christine, and I'd like to add in terms of the overall vision, so within the context of the milestones as you progress through the challenge, is that it's the reduction to practice. So really the ultimate goal is practice, adoption, implementation. So the real world application of some of these technologies.
Excellent. Thank you. Thank you for that answer. It was very thorough. Thank you. All right, next question. For the NAM platforms previously supported by NIH funding, can those NAM systems be qualified for further improvement in this challenge?
Yes, my understanding is that federal funds can be used. So in the research projects that have been funded thus far, those types of projects can be considered for this challenge. However, it needs to be the funding. So there's additional information provided in the challenge in terms of the language for the legal aspects of this, but those projects can be refined and further developed so that, and in fact, but however, as again, with the challenge, it's a different type of mechanism. It needs to be a completed product to be considered for the challenge, and you can't get funds for something that you've already accomplished. So that you've already received federal funds for. So it's a complex answer. I think we will also develop further answer in the FAQs to make sure that we point you to the right section in the challenge announcement.
Excellent. All right. Is addressing drug toxicity using combinatorial NAMs a priority for this challenge?
Yes, it is one of the priority areas, toxicity testing.
All right. Next question. Can you give a couple of examples of specific contexts of use related to priority areas?
I think that's a little bit of a difficult question because the context of use is really going to be very dependent on the NAM. I mean, it could be things such as drug-induced liver injury. There can be toxicity related contexts of use. There's really a very large range of what the potential context of use could be.
Excellent. Thank you. All right, next question. How is scale up defined for phase 2, milestone 1?
So the requirements for the later phases for phase 2 and phase 3 will be announced on the challenge website as we get closer to those stages. So more information and more specifics about those requirements will be coming later, including the evaluation criteria for those phases as well.
Excellent. Thank you. Yeah, and please feel free to follow the challenge and sign up, and you will get challenge updates. So you'll see when those updates come through on the challenge page. All right, next question. Can a combination of AI and OOC organ chips be a solution for applying to this challenge?
Yes,
Yes.
that would be. Mhm.
Excellent. Next question. Primary human cells often vary in their performance in complex in vitro models. How do you envision the validation stages taking this into account?
Um, I think that we completely agree. Validation of those types of primary cells can be very difficult. Fortunately, the VQN has amassed a number of both public and private entities that have partnered with NIH on this effort, and the swath or the breadth of the expertise of that group is really enormous. So I think that we will have a lot of input and expertise when we get to that point. I know some groups find that they switch to like iPSCs versus primary cells because for that exact reason, because of variability. I think that's something that we'll be assessing though on a case by case basis as the projects move through the challenge competition.
Great. All right. I'm not seeing any further questions coming through. Oh, it looks like there might be just, okay, here we do have another one. Okay, just give me a moment to read it here. Okay, so the combinatorial NAM has to be the integration of two categories of NAM techniques. What about combining two organoid types for co-culture? Does this qualify for combinatorial NAM?
I think that could potentially be two different in vitro DIMs, so combining into one modality, so that could potentially be feasible.
Excellent. Okay. Questions are hopefully coming in. We'll just give it a minute, and it's a reminder to the solver group. Please do type your questions into the Q&A box, and we'll be sure to ask them to the experts here. We'll just leave it a minute to see if any further questions come in, and we'll get those out to the group. So we'll just hold tight for a minute here. Just a reminder to those on the call, if you do have questions that come up following the Q&A webinar, please feel free to submit them to the email you see on your screen, or utilize the forum section on the challenge page, and we'll be sure to get answers back to you. All right. Another question here. Is it important to build from multiple donor sources?
I think that's really going to depend on the model. It so I think that reproducibility and validation are really the focus of this challenge, as Margaret mentioned, that it's not just the technology development of these NAMs, but it's more of the implementation and adoption and use of the NAMs is what we're trying to move forward with. So it's really going to be what is most reasonable and makes the most sense, and what is going to allow the technology to be sufficiently reproducible for whatever the specific context of use is. There are no requirements for that, so it's really going to be up to the solver.
All right, great. And we'll just give the group another minute or two to submit some final questions before we wrap up for today. Just sit tight for a moment and see if any further questions come in. All right, we're not seeing any questions come in. Hopefully, we've covered off a lot of your pressing questions here, but if they do come up, please, as I've mentioned, either email them to the email you see on your screen or add them to the forum section on the challenge page. We do want to thank you all for taking the time to join us today. Please do keep in mind the submission deadline is on March 1st. And again, we will be uploading a recording of today's webinar to the challenge page. Thank you all for your time and have a great day.